Maybe let's get started. Well, good morning, everyone. Let's welcome to the Power Talk webinar. We have great speakers, and it's really my pleasure to introduce my new friend and colleagues from Common Fund Data Ecosystem, Professor Sumner, with us today. Dr. Sumner is a professor of nutrition at the University of North Carolina at Chapel Hill's Nutrition Research Institute. She directs the Metabolomics and Exposome Laboratory. Uh, and that this is a really fascinating field. And for those of you who went to the ADIS meeting and hear Professor Mike Snyder's talk on just tracking glucose and responds to a few food item and personalize it. And if you're fascinated, and then imagine that you have to track thousands of metabolites, and then also figure out that what the environment may have impact on those things, and keep track of longitudinally over many people. And this is tremendous skill and tremendous data. That's what she's really good at. I think that will certainly the research in this direction she will share with us will really advance the personalized health through precision nutrition. She has lots and lots of funding from the NIH and Nutrition for Precision Health Consortium. And we have lots of interest at UAB to also get access to data like that. And in fact, I'm just a baby in this field, and we have Steve Barnes, and who've been working with her as colleagues for many decades. And so we're so excited to have you here and talk to us about the opportunities and hopefully build some ties. Uh, Go ahead.
Okay, thank you. Thanks for the introduction and the opportunity to speak with everybody here today. And yeah, I've really appreciated all the opportunities that I've had working with Dr. Barnes and in that phase one NIH Common Fund program. And I think our intersections have gone way back from there also. My husband, who works at RTI International, tells stories of all the problems that he's called uh, Dr. Barnes to help him solve over the years. So every once in a while, he'll go through a long list and is very appreciative of that. Radiochemical, radio label, all kind of things. So yeah. So any rate, so what I wanted to talk about today is sort of this untargeted metabolomics and what does it mean to be doing untargeted metabolomics and what can you use it for? And particularly what my interest is in is the development of intervention strategies. So what kind of interventions can we develop with untargeted metabolomics? So I do have a keen interest in pharmaceuticals and drug development and identification of drug targets, but I also have an interest in nutrition. Um, and so that's going to lead me to first talk about as nutrition as part of the exposome. So we all know that we do have some nutrients that are essential, and they're essential because they're compounds that are supplied from our foods that can't be synthesized by the body or cannot be synthesized in the amount that's needed. And so that's why they're referred to as essential. And they're needed for normal growth and development and reproduction. And these nutrients provide energy. They serve as cofactors for hundreds of our biochemical reactions of our host metabolism. So get that host metabolism going like you want to. You got to have these essential nutrients. They do serve as antioxidants, scavenging free radicals, for example, out of cell membranes. They're involved in gene transcription and in DNA methylation, and they provide structural constituents for the formation of everything in our body, our cells, our bones, our teeth, in our blood. So when we're looking at metabolomics, we really have this opportunity to also make sure that we're examining these essential nutrients carefully. So I'll be talking about some of that in studies. Now, nutrition, we know plays a role in mitigating the onset or progression of disease from, many very good epidemiology studies that have linked nutrition or nutrients with specific diseases. But we don't really know how to prescribe nutrition yet. Maybe we will someday. What we do know is that optimal levels of nutrients can depend on our genetics, lifestyle factors, life stages, states of health and wellness, and then exposures. And these exposures can be to food and beverage components, the good things in those. They also can be additives and impurities in those foods, drugs, medications, and other sources of environmentally relevant exposures and pollutants. And so when we're thinking about, our health, it's nice to try to think about all of those things together and what are our exposures, or what is our exposome, but then what are we going to be able to do about that? And that's when essential nutrients come in. So this is just a concept slide of the exposome in precision health. And the idea is that exposome can inform precision health through understanding how exposures and perturbations in endogenous metabolism are linked to our genetics and to our states of health and wellness. And so our environment, we have all kinds of exposures, whether it's tobacco use, e-cigarettes, chemicals in the household and workplace, in our personal hygiene products, or our nutrients, our dietary intake, etc. So drugs, medications. And so these influence our metabotype, as well as genetics, polymorphism, stress, family structure, so there's all these things that go into influencing what our metabotype looks like. So when we look at these metabolic perturbations in our metabotype, we can look for biomarkers that associate with a wide range of disease states that have been investigated, some in our lab, many, in laboratories. We can look for biomarkers that associate with, the onset of diabetes, or cognitive development, or cognitive decline, or pregnancy complications. But then also we can look at those metabolic perturbations in a different way to understand mechanisms that can relate mechanisms that can provide us information about pharmacological targets or nutritional targets, and can inform us about the intervention of diets, nutrients, microbes that are important, or even environmentally related exposures that we may want to reduce, okay? Or what could we do about those? Okay, so there you go. So one of the technologies that we've worked on over the years is using UHPLC, Q-Exactive HFX, Orbitrap mass spectrometry platform. And we've used that platform to detect 10,000 to 40,000 features in biospecimens like urine, stool plasma, seminal plasma, blood spots, sweat, hair, even, tissue extracts. So it's a wide range of biospecimens that we can examine and have examined in our laboratory and others. So we have all these features, so what are we going to do about that? So one thing we do is we take compounds and we extract those and run those on that same platform. And we develop what we call an in-house physical standards library. And in this case, we have some 6,000 compounds that we've run on our platform. And these are metabolites of host and microbial metabolism, and many types of exposures that I'll talk more about on an upcoming slide. But even if we run some 6,000 compounds, that's not necessarily going to assign all of those peaks. So we've worked with, this is a collaboration that we've had ongoing between RTI International, where I used to work, and also UNC Charlotte with Susha Du. So RTI with Tim Fennell and UNC Charlotte with Susha Du, to develop a computational metabolomics framework And we've developed an open source software called ADAPT for automated data analytics and processing. And so what we do is we use that software for peak picking and processing that data, extracting analyte information, and then we can we have algorithms that we match signals to that in-house physical standards library using retention time, exact mass, and MSMS. And then for the signals that don't match to the library, we also have rapid algorithms to match signals to over 1.7 million spectra that are in public databases. So we have a chance to get at those things that have these good matches to our in-house library. And then we can provide many, many annotations to public databases as well. So that's kind of the framework of how we're capturing data in most of our studies. I do want to mention that Tim Fennell at RTI, who is my husband, and I'm supposed to disclose that in any presentations that I give. That's my university says I have to do that. So I know I feel kind of weird about that, but I'm a rule follower. So Tim's my husband. He does have a GC exactive method that he's using. My laboratory focuses on this UHPLC method. So, and his lab does most of the GC exactive. Okay, so just I recognize that we're not going to get every metabolite detected on our UHPLC untargeted platform. So we can go to other platforms as well if we want more. But we get so much information with one platform, it takes us so much time to dig through that I'm not usually looking to get more. Okay, so what we're doing is we're looking at host and microbial metabolism. We know, so the thing I want to emphasize is we know the range of analytes that we detect on our platform because we've run standards that span across many different kinds of compound classes. And so those are kind of categorized here as host and microbial metabolism, like our amino acids, bases, nucleotides, sugars, fatty acids, lipids, bile acids, hormones, etc., following into that host metabolism and microbial metabolism. And then we know that we detect a wide range of environmentally relevant metabolites because we've run metabolites of like PFAS and parabens and phthalates and volatile organic compounds and flame retardants and pesticides and herbicides. And so we know that we're detecting all these classes that are listed here. And we selected those classes because we know that in NHANES studies that many of these compounds or their metabolites had been detected in individuals' biospecimens, across the United States. And then also we've acquired data for the EPA ToxCast library as well, which is the priority list of chemicals. So we have a good wide range of environmentally relevant chemicals. And some of those are in the foods that we eat, of course, and in the beverages that we drink. So they're not just, breathing into our lungs, but we're actually putting them into our bodies by consuming foods and beverages. We have a lot of drugs and medications on that platform as well. And then we have standards of metabolites that are related to the ingestion of foods. And we know they're related to the ingestion of foods because these metabolites have increased in individuals who consume diets that were high in polyphenolic compounds. And those studies have been run by Dr. Colin Kay, who's one of our collaborators on this project. And so we took those analytes detected by his targeted methods and put those on our platform as well. And last, I just want to mention that we detect a wide range of vitamins and vitamin-like compounds, metabolites of pretty much all of the B vitamins, choline, involved in choline metabolism, folate metabolism, metabolites of vitamin A and vitamin D. We detect some of the phyloquinones. So there's a wide range of metabolites that are detected, as well as other essentials or vitamin-like compounds, carnitines, essential amino acids, and our omegas. So we see this stuff simultaneously. So since we're seeing it simultaneously, we should be using it simultaneously. Yet many investigators only look at like one classification of exposure and one outcome, and that's still happening today. But I'm trying to preach the gospel of using all this together, right? So this is one of the studies that we did with Emily Harville at Tulane University to examine what I'm going to call the internal exposome and pregnancy complications. And this is internal exposome limited to the detection by our UHPLC MS platform. So we assessed first trimester serum samples from individuals who had an uncomplicated term birth or had a hypertensive disorder of pregnancy or a preterm birth. Now, at the time that these samples were collected, they didn't have any signs that they were going to have those complications, right? So they looked clinically healthy individuals at that time. But some of them went on later to have these hypertensive disorders, preterm birth, or uncomplicated term births. So we compared those different cases, case controls, the control being the uncomplicated term birth, and we compared those using our untargeted method. And what we found is that there were endogenous and exogenous metabolites that significantly differentiated the cases and the controls using our data. And so those could be early biomarkers for the early detection of these pregnancy complications, right? At least could inform the doctor there's something that could be going on, right? That you're not able to detect yet in that first trimester, but we know by metabolomics something's going on. Now, we did develop pregnancy complication risk models, and they had greater predictive value when including both endogenous and exogenous metabolites. And I just want to reiterate, by endogenous, I mean those that are produced by our host metabolism, and exogenous, I mean those that are produced because we have eaten or been exposed to something that got into our bodies somehow and produce these exogenous metabolites. Okay, so they were more predictive by combining those. So this is just a summary slide and certainly doesn't have all 10,000 of the metabolites on it, but it gives you a little view of the way that we were seeing the data. So one of the things that was really sort of interesting about the data is if you look at the yellow box towards the center, or maybe that's tan, we saw one compound that's a metabolite of butylated hydroxyanisole, which is a widely used food additive, and that showed a strong correlation with gestational hypertension, and this was done with multiple regression modeling. Some BHA metabolites have been found to induce cellular DNA damage, and the chemical BHA was placed on the European watch list back in 2015, but is still used in the United States today. So that's one thing I mean about informing us about exposure reduction. So here we have something that is a known food additive that has a strong correlation with our gestational hypertension, but has been known for many years that it induces cellular DNA damage. And that was sort of interesting to see that in our metabolomic study. And I hope that these kind of studies can help influence policy. Now, there were many other exogenous compounds you can see at the top left of the slide that significantly differed between our cases and controls, including metabolites of bisphenols, parabens, volatile organic compounds, medications, and tobacco-related analytes. And none of those we would want to significantly differentiate case and controls because it would be better not to have them in there anyway. But if you look over to the top right box in the lighter blue, you can see some of those, like monohexyl phthalate, had a strong correlation with hypertensive disorders of pregnancy, and other compounds correlated with preeclampsia or volatile organic compounds. So looking in the same data set, we can see these kind of correlations for these host metabolism. And then down at the bottom, you see we looked at endogenous pathways, and we had the endogenous pathways correlated with preeclampsia included the androgen steroid hormone metabolism, and then we had other pathways that correlated with preterm birth, like threonine or carnitine-related pathways with hypertensive disorders of pregnancy. So this just gives the idea that we're being able to look at these exposure things and their correlations, as well as looking at host metabolism correlations with pathway perturbations. And then this whole concept that the modeling is stronger by looking at both of those together. Now I'm going to move on and tell you about a study with osteoarthritis because I wanted to give several snapshots. This is a study that we've been doing with Richard Lozier and Amanda Nelson, who's at the UNC Thurston Arthritis Center, I guess it's called. And in this study, we looked at both 16S RNA as well as our untargeted metabolomics, and examined stool samples that had been collected from individuals who had been diagnosed with osteoarthritis and compared those with stool samples from individuals who had been diagnosed as not having osteoarthritis. So that's what's been meant by case and control in this case. So, what we saw when we compared our untargeted metabolomics profiles is we saw perturbations in leukotriene metabolism, and that's related to the precursor polyunsaturated fatty acids like omega-3s. We saw perturbations in fatty acid synthesis and in tryptophan metabolism. We saw a decrease in short-chain fatty acids in the OA group versus the non-OA group. And you know that propionate is produced by colon microbes like Clostridia, and they're related to the amount of dietary fiber that one consumes. Lower Clostridia was associated with OA in our microbiome data as well. Short-chain fatty acids are known to regulate energy utilization, like the fatty acid metabolism, where we saw perturbations in fatty acid synthesis. They are involved in inflammation. And short-chain fatty acids are used in some countries to mitigate inflammation and are used in patients who have arthritis. We also, but they were not being used in the individuals who participated in this study, I wanted to say. We saw a decrease in N-acetyl-D-glucosamine, which is a natural compound in cartilage, and it's used in the US and elsewhere as a supplement for joint pain, inflammation, and used in inflammatory bowel diseases. And some studies have shown that this compound protects the lining of the stomach and gut. Now, in our study, the individuals were not using glucosamine, and it was decreased in those individuals with OA. And so were the short-chain fatty acids decreased in our study with those individuals. And again, they're produced by dietary fiber by colon microbes, and those particular microbes were also decreased using our microbiome data. So if we put this together, and I know that it comes together a little bit rapidly in this presentation, but there is a publication on this already, that what we propose from looking at these metabolic perturbations is that we would have individuals have diets that have a higher omega-3 ratio to omega-6, for example. So increase that omega-3 in the diet, increase fiber over protein to be able to produce those short-chain fatty acids. And then one could supplement with short-chain fatty acids as well as with glucosamine. So this is sort of an example of what I mean about trying to formulate a dietary intervention or recommendation based on looking at these perturbations. Now, but what about exposures? So I really like exposures, right? So no, I actually don't like exposures. We don't like them, but I like to think about them when I'm looking at the data. Okay, so this is a snapshot of one analysis. So since we had some 53 environmental compounds that we saw in these stool samples, matching to our in-house physical standards library, okay? So there were many other ones that we had annotations for to public databases, but there were at least 53 that matched to our library, which is pretty good to be looking at these 53 simultaneously. So we use correlation analysis of these metabolites to show connectivity between environmental relevant metabolites, inflammation markers, and endogenous and microbial metabolism. So I'm going to tell you one example. It's kind of complicated, so I'll talk about it and then I'll go over it a little bit again. So we saw a peak that matched to an insecticide that correlated with an herbicide. So that was very interesting. It had a very strong correlation between this insecticide and this herbicide. And I wondered if that was indicating a potential co-exposure in ingestion of foods with those on there. Could be. I don't know why, just an idea. Okay, we also saw that that peak to that insecticide also correlated with a polyphenolic compound called hydroxycinnamonic acid, which is abundant in fruits and vegetables and may be that source of exposure. Okay, it could also be produced endogenously, I guess, but just building on the story. Now, that insecticide also correlated with gut microbes from a phylum that produce short chain fatty acids. And on that last slide, I just told you how our short chain fatty acids were decreased in our study. And so it occurs to me that perhaps this insecticide is targeting those microbes and causing them to have a lower production of short chain fatty acids. So this would be the idea about identifying microbes as a target, right? Okay, so and so then we saw a correlation between this insecticide with neurotransmitter related metabolites. And these metabolites are products of tryptophan from that's formed in the gut. So we saw decreases in neurotransmitter related metabolites that are produced from tryptophan, and tryptophan is produced in the gut. So it seems like this insecticide could be targeting that pathway that produces tryptophan, causing decreases in these neurotransmitter related metabolites. So that's kind of interesting how that fit together. And then we saw several metabolites that are associated with mitochondrial fatty acid beta oxidation that were increased in individuals with they were decreased in individuals with OA, and mitochondrial fatty beta acid oxidation has been proposed as a target for the development of osteoarthritis. So that's sort of when I say looking at this power of this untargeted metabolomics. We just now have 1, 2, 3, 4, 5 hypotheses that we can have, people work on to look at OA from just this one correlation of this one insecticide. And of course, there were many other environmental analytes that we could do the same kind of analysis and look at these correlations on. It's a good story, right? So, yeah. All right. So, I wanted to just go through this example, and I want to give us time to talk. So I'm not going to go through all my slides today because I realize I am talking a little bit more detail or slower than I thought I would go. But anyway, so I wanted to talk about this study. When I started this study, it's a collaboration with the National Institute of Drug Abuse and investigators at the University of Tehran. And yes, this was all approved by all the official systems to do this. This study involved originally to develop objective biomarkers for opium use disorder, but during the conduct of the study, it became clear to me that, wow, we could also develop nutritional intervention recommendations. And so that's kind of what got my attention, although I do think biomarkers of opium use disorder are incredibly important. So that's what the study's about. What we did is we obtained urine samples from individuals who reported opium use and obtained urine samples from individuals who reported never using opium. These samples came from a region in Iran where telling the truth about opium use would be just fine. So we're pretty sure that those reports are accurate. And in this particular case, we compared the metabolomics profiles of the urine from individuals who reported never using opium with those who reported using a lot of opium. And I'm going to say, equivalent to smoking a pack a day of cigarettes is sort of a lot a day. So, okay. And then we also compared the urine samples of individuals who were opium users who were diagnosed as OUD positive, compared with people who were opium users who were diagnosed as OUD negative. So we're not just saying case and control, we're saying diagnosed as OUD positive and diagnosed as OUD negative. So we know that OUD negative diagnosis was there. We saw many differentiating features that could tell us what are the perturbations that are induced by opium exposures, and then what are the metabolites that define an OUD positive diagnosis. So that's what we set out to do. And we got many interesting findings. And I see that a lot's information's here, and I want to start in the upper left and tell you that in opium users, we saw metabolites derived from opium use. Check. We had the data going in the right direction, right? Now, we also saw many tobacco-related analytes that were increased in opium users, which doesn't surprise us because the tobacco use was higher among the opium users, okay? And that's not surprising at all. Now, looking down towards the bottom of the screen, we saw environmentally relevant analytes that were higher in opium users compared with the non-users, and this included phthalates that could be derived from the plastic tubing that's used in the opium intake, and they were much higher, like the p-value was to the -8th, right? And these phthalates are important because they have been implicated in obesity, diabetes, learning, and cognition as well. And learning and cognition is sort of one of the OUD diagnoses. Keep that in mind. Now, we also saw metabolites that are known to be derived from parent compounds that are formed during the curing and combustion of plants. And one of those was acrylamide metabolites. And they were so much higher. It was 10 to the -21st in p-value, higher in those opium users compared with the non-users. In our samples, people were not diagnosed with any specific types of diseases, but it's known that cancer rates are higher in this cohort of opium users. And it could be due to these higher levels of toxins that are produced on the curing or combustion of that plant matter. And then up in the upper right-hand box, I want to say we saw perturbations in neurotransmitter metabolism, Krebs cycle metabolism, 1-carbon metabolism, glucogenesis, lipid metabolism, and essential nutrients. I'm not going to tell you about all of them, but I'm going to tell you about a couple that I think will interest you. So this is the perturbations in neurotransmitters. And I just wanted to point out that we saw a metabolic disruption in compounds like tyrosine, tryptophan, serotonin, kynurenine, and phenylalanine as well. We looked at the perturbations and B vitamins, and B2 and B6 were very significantly different between opium users and non-opium users. And guess what? They're required as cofactors to produce to make dopamine from tryptophan or serotonin, dopamine for tyrosine or serotonin from tryptophan, and then B6 and B2 are both required to produce the 3-hydroxykynurenine. So the point here is that with reduced levels or competing for absorption or transport, our opium users may not be producing as much of some of these metabolites in the neurotransmitter pathway because of lower relative levels of these B vitamins. Not necessarily from intake, it could be from transport mechanisms as well. So we got to keep that in mind. And then just to say that OUD positive opium users versus OUD negative opium users also show differences in metabolites on these neurotransmitter pathways. Okay, now we're going to just look at sugar metabolism and Krebs cycle metabolism, and just to give some examples that we saw perturbations in inositol, fucose, sucrose, mannose, citrate, cisaconate, sucsate, malonate, and that's not all of them, but it's some of them. Now, this is sort of interesting because it takes 4 B vitamins to produce acetyl-CoA from pyruvate, and 3 of those B vitamins, B2, B3, and B5, were greatly perturbed between opium users and non-users. And it takes 7 5B vitamins to convert alpha-ketobutyrate to succinyl-CoA, and 4 for alpha-ketoglutarate to succinyl-CoA. Now, so with our B vitamin perturbations that we see, it's not surprising at all that we would see perturbations in Krebs cycle metabolism. Now, this can come really important also because we know that B vitamins are needed to produce, well, actually, from the Krebs cycle, we produce the NADH and the FADH2 associated with those vitamins that go into the electron transport chain. And once in the electron transport chain, we're using B2, B3, cytochromes, our iron is in our cytochromes, etc. All that's going to produce ATP. So if you're having a general metabolic disruption in these cycles, could be resulting in less ATP, and that could be related to an OUD diagnosis as well. And we did, when we were looking at opium users versus non-opium users, no, this is OUD positive versus OUD negative opium users. There were metabolites from sugar and Krebs cycle metabolism that differed between those as well. Okay, the last one I'm just going to mention is perturbations in 1-carbon metabolism. And we saw perturbations in choline, phosphorylcholine, betaine, methionine, threonine, etc. They're listed there. I can't even see well enough to see them all, but you can read them. But at any rate, we know that low levels of choline are associated with cognitive and memory disorders, and that could be related to an OUD positive diagnosis as well. And we did see those perturbations between OUD positive opium users and OUD negative opium users, including phosphorylcholine. And so knowing that these are related to memory disorders and mood disorders, and then knowing that they're decreased in those OUD positive opium users is important. Also, you know that choline is metabolized to acetylcholine, and that's a precursor for the neurotransmitters and could very well be associated with some of those neurotransmitter perturbations as well. I did want to mention in this area in Iran that foods are not fortified with these B vitamins. So individuals may not be getting enough of these B vitamins or enough that can mitigate their exposures to opium. Okay. Now, they do have prenatal vitamins, but they don't have any foods fortified in this region in Iran. Okay, so this just led to sort of this hypothesis that we could be using this untargeted metabolomics to derive nutrient cocktails composed of, you know, whatever our essential nutrients are that we're seeing perturbed in our metabolic pathways to be able to mitigate, and in this case, mitigate against addiction or other side effects of addiction. As well, our goal is to be able to identify biomarkers unique to an OUD positive diagnosis. We did find many in this study. Of course, that would need to be validated. But I did want to say that a lot of these times when people use another cohort to validate something, that they don't validate it and they say, oh, those biomarkers must not be good, but it could be the genetics of the cohort. And so I think we need to be really careful about throwing away biomarkers and saying that they're not good because we can't correlate them, or we can't use them in a totally different cohort that may, you know, have different genetics that are related to these metabolism. As a last point, we did find exposures to environmentally relevant compounds such as phthalates and metabolites that can be derived from the combustion of plant matter that could exacerbate the opium use. And it could be related to the higher incidence of the cancer, the cardiovascular disease, and diabetes in this cohort, as well as to memory and cognition. And again, in our samples, individuals did not have any disease diagnosis other than the opium use. Okay, so that's it. And I have a lot of other stuff to talk about, but I have 15 minutes, and I'd rather take the time to just have a discussion. If anybody would like to ask some things, and if not, I'll talk about another study. Right? What would you like to do, Jake?
Yeah, that's a fantastic talk, and I'm glad that you leave a lot of the time for discussion. I'm so excited. I'm pretty sure if we give the mic to Steve, and then he's gonna use half an hour and more. So I'm gonna grab the mic first. So sorry, Steve, and I'm gonna ask some questions first, since I invited Susan, and then yeah, we'll give the mic to audience. So first thing, I'm really just so happy to get some everyday nutrition knowledge. In fact, when you talk about the protective values of fish oil and glucosamine, I for the first time I actually run to my nutrition supplement cabinet. I took some pills. And then I thought, oh, when you talk about the opium production with a B6 and B2, I thought I would take some multi supplement. And so there's a wealth of knowledge that you guys have. And for people outside of your field, we felt that we're really poorly educated. But my actually question is actually twofold. One is that it seems that the nutrition is so complex, and when we try to study nutrition, and how do you actually go from single factor causal analysis, which you have done a fabulous work, to something multifactorial, which will be extremely difficult to establish the causal relationship, right? That's one part. And another challenging part is actually to how do you practice this precision nutrition? Uh, I remember Mike Snyder mentioned that he tested the subject with a different food item, cookie, cereal, and so on. There are people who are actually their glucose level do not increase, spike when you give them lots of cookie, and then there are people who actually spike. And then I was thinking that for nutrition, for even the same measure of good or bad nutritional items or metabolites, then multifactorial, it is so complex for us to, for the regular person to think about what is the precision nutrition and how can we do? And I got really scared when I saw all of the list of contaminants. I have it in my kitchen all the time. So does it mean that I cannot eat anything, or do you have some better tools? So let's start with that.
Yeah, so I like having 50 questions at once because that sort of helps me focus. Um, So, so first of all, the causal relationship. So that becomes very important, and that's one of the reasons why, we're trying to detect as many things as possible, simultaneously, and then look at the correlation between, for example, a food component and all these other things that we measure, or between an environmentally relevant compound and all these other things. We can also look at classes of environmentally relevant compounds and look at them sort of additive rather than just one at a time. Or we can look at multiple compounds across classes that people will be most likely to be exposed to. So trying to get to what is the causal relationship? Is it coming from, chemicals in our environment and our foods, or is it coming from, um, you know, uh, genetics, of course, because genetics is involved in how you're going to process those foods, right? And what are you going to be producing from the gut? So this whole idea of using the multiomics and getting as much as you can is very important. I'm going to get back to that later, but I wanted to try to address another one of your questions. Okay, so so one of the things that we're doing with the Nutrition for Precision Health program sort of gets to this idea of, how are we going to be able to prescribe nutrition? What's it going to take? So in this program, which is headed by the NIH Common Fund, what we're doing in my laboratory is looking at the metabolic profiles, which includes untargeted analysis plus quantitative profiling of polyphenolic compounds and detailed profiling of host metabolism, so and then many clinical assays. And then we're gathering that data together with GWAS data, microbiome data, and that's from individuals across the United States, okay, and a very large scale study of some, let's just say, 8,000 or so participants who were on their baseline diet, and then they get a meal challenge, and we know how they respond before and after that meal challenge. And then they get prescribed diets, nutrition for precision health diets. And they get those diets in a randomized crossover clinical trial, and with the washout period between diets. And at the end of each diet, get a meal challenge. So now in this case, we're getting more data, big data, okay, to look at how do individuals respond to a meal challenge, like a liquid meal tolerance test, when they're on their habitual diet, their normal diet, and how do they respond after they're on diets that are formulated, say, to be like a Mediterranean diet, or maybe like a healthy high-fat diet, or a traditional, say, diet of hamburgers and hot dogs, right? So we get a better idea of how individuals respond to that. So in this study, what we're doing is using clinical assays as part of our phenotypic anchor, because like Jake, you mentioned that people can, their glucose can be different depending on, how they respond to eating a cookie. So we're looking at all of these clinical assays listed here and how individuals respond to that liquid meal tolerance test after they've been on their habitual diet or any of these prescribed diets. And then using AI modeling, okay, which is being done by a group at West Point headed up by Di Thomas, to use AI modeling to take all this data together from GWAS, microbiome, phytochemicals, untargeted metabolomics, et cetera, behavioral phenotypes, physical measures, everything, and to say, how does that all come together to talk about how individuals response, right? To be able to predict an individual's response to dietary intake. Okay, so I think that might answer your question a little bit, sort of at a high level. And I did want to say this study is really ideal for doing that because we do have a very large sample size of over 8,000 participants. We're trying to ensure representation of the diverse US population, and that this should be able to help us inform diets that are most appropriate for health outcomes that are linked to subpopulations, as well as provide objective biomarkers for things like dietary intake, behaviors, energy expenditure, et cetera, that we may get those objective biomarkers at a subpopulation level rather than trying to have a one-size-fits-all kind of biomarker. So I hope that answered some of the 50 questions, or that I caught on to something that you were saying appropriately. Okay. I also wanted to mention that in the United States, we have so much fortification in our foods already, right? So if you look on, just I'm sure a lot of you probably read on the bottles what what foods are fortified with. And so most people, are probably getting a lot of vitamins through that fortification, and some of them may even have too much. So, for example, folate um, may some of us believe that folate could be too much fortification, especially for children in the United States. And so I'm really happy to see that. I was actually very happy to hear RFK Jr. talk about nutrition and really trying to get a better understanding of what people need, because we could be over fortifying some of our nutrients in foods, and that could be linked, for example, to increased cancer risk. So folate would be one of those of a huge concern. If you're not getting enough folate, there's a link to increased cancer risk. But then getting too much folate seems there's also increase in cancer risk. And so determining exactly what is that optimal level of folate that people should be getting and be getting by age. It may not be the same across all ages. And so this one size fits all fortification may not be right. And we really need to do a lot more research on that. And I really do appreciate him coming in and being, provocative and shaking that world up a bit, because I think it needs to be shook up. Yeah. Yeah. There you go.
Right. I'll give Steve some chance to ask questions.
Yeah, sorry.
Yeah. Well, you're right. I could talk for an hour, but I'm going to ask one very specific question to somehow bring this to the interests here at UAB in data science, which is to the electronic health record. And so, in the studies you've done, have you been able to pick out the medications that the patients or the subjects were taking? And did that correlate with information that was given to you either by the patient or by the medical team that might have been behind that patient? I've
Yeah.
seen amazing discrepancies here at UAB, but I'd value your experience.
Yeah. So, yeah, so let me try to focus my thoughts on that, because that's such a great question, so open-ended. So, in the answer, so I could talk an hour about it. I want to narrow it down to a few seconds. Okay. So yes, we do see uh um medications. We do see metabolites that would be derived from, example, either first or secondhand tobacco use. We also see metabolites of illicit drugs that are not reported. And we see these things simultaneously, right? Now, some of those reported, some of them aren't reported. Um, when we design studies to look at predictive uh, uh, markers for, say, early detection of disease or disease progression, et cetera, usually we're getting biospecimens that are collected before individuals are diagnosed and put on a drug or medication. And so that doesn't complicate our, because we're looking at that prediction, right? And they don't have those now. But some studies we do is after people have been diagnosed and they are already taking the drug or medication, some sort of medication after that diagnosis. And so now we're looking at the drug metabolism as well as the progression of the disease. And so in this case, what we're looking more at is responders and non-responders to treatment, and how we ask the question of the metabolomics data. And then can we tell if somebody is within our data is a better drug metabolizer, and that's why they had a better response. And then how does that link to host metabolism? Okay, so we can actually look at, say, pathway enrichment analysis on the drug or on the metabolite, how it perturbs host metabolism by looking at that pathway enrichment analysis to be able to say when somebody is metabolizes the drug differently, or maybe they look like they're metabolizing it faster, how does that link to those perturbations in host metabolism? So we are looking at that quite a bit. Yeah, but we do see things that aren't reported for sure. Yeah. And that becomes sort of complicated because some states, like, for example, if you see an illicit drug in the data, and say if it's from a pregnancy cohort, they're supposed to report that. So there is a lot of ethics complications coming up with metabolomics. We always ask people up front, if we see illicit drugs, what are you going to do with that information so that, they can kind of prepare for that. Yeah. 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 But, yeah. Yeah.
So
Okay.
it's an amazing the amount of data. And of course, what NIH is now doing and requiring, and I guess this is what Jake is associated with, is making sure that all the data the raw data we collect goes into a depository. So down the road, the data scientists can take access to this and perhaps find things that you and I can't even see, but the computers can start to pick out.
Yeah, so this is true. So so that sort of opens a can of worms. So for most all the studies I work on, the data is called publicly accessible under FAIR principles. And that's what is said. Like for the Nutrition for Precision Health study, the data is being made publicly accessible through the All of Us workbench. Okay. I'm also doing another ancillary study to All of Us, which is a diabetes study, and that data is being made publicly available through All of Us. Those are some of the ways, and you see all the list of things on this slide. That's a discovery science resource that all of you will be able to use, and it's going to have all of this data on it, including access to the raw data. So Steve, my raw data from untargeted isn't going to be on the NPH workbench, but there'll be a link to access it. So it'll be accessible to anybody who wants to start with raw data, as well as various formats of the pre-processed data. Yeah, but all of this data is going to be there. And some of the data is already, some of this Nutrition for Precision Health study data is already on this workbench, but not released to the public yet. We're just getting it there and starting to look at it and make sure everything looks all right. Yeah, but it's coming. Yeah. Yeah.
So a very quick question. So it's about the drug discovery. We know that that many drugs can be inspired by nutritional studies, but I wonder if you know of databases and research trend on nutraceutical compound development. Are they on the rise, stalemailed, or just out of favor now? And if so, I'd like to maybe find someone to collaborate. Like for example, the flavonoids from fruits and onions and have been made into, I don't know whether it's a require FDA approval for those, but it could be enhanced uh, nutritional supplement or it could be drugs. Do you know much about these?
So yeah, I'm not sure what your question is, so I apologize for that. But yeah, I mean, people are really interested in nutraceuticals and development that polyphenol compounds has a huge interest if and even ingestion of cranberries, ingestion of blueberries, and we've done some of those studies as well with looking at individuals' response to we have a recent paper individual's response to ingestion of cranberries, right? And what's in those cranberries that's causing that. So yeah.
My question is more about has it already been systematically studied? So people are already scanned through everything, or there's lots of opportunity for drug discovery for these nutraceuticals with potential intellectual property.
I think there's lots of opportunities for study. Yes, I believe that that would be true, that it's wide open, lots of opportunities for study. And I think that in a study like this Nutrition for Precision Health data, you should be able to get a wealth of information out of that because they have the dietary assessments for each individual person, and it will also record any supplements that people are taking or any other kind of natural products or anything like that. There's probably going to be a lot of stuff in there that could be used for that exploration. So I think that field's wide open. Now, one of the things that I'm sort of more interested in is how do we take our data to be able to formulate a nutritional cocktail or a diet that would augment the positive response or the beneficial responses of drug treatment and mitigate against adverse side effects or potential adverse side effects of drug treatment so that one can lower the dose of those drugs by using nutrition to augment them, and then that will mitigate those side effects as well. So that's, I think, a wide open area to investigate, and I think untargeted metabolomics is just ideal for that. Uh, yeah, yeah.
Susan, the NIH just had a botanicals research center program going. Uh, Connie Weaver and I had support for 10 years from 2000 to 2010. But there was a guy at Yale, Chinese guy, who found that by giving some of the cocktails that are available in Chinese medicine, massively reduce the dose level needed for anti-cancer drugs, so much so that the intended effect was maintained, but all the side effects disappeared.
There you go.
Yeah.
Perfect. Yeah.
Yeah. So there's lots of people out there, Jake, just, now you're coming into this area
Yeah.
that Susan and I can introduce you to.
Yeah.
Yeah,
Um,
happy to.
Yeah,
I've
that would be, that's a
Yeah.
really good example. And of course, grapefruit's a really good example for lipid lowering drugs, and you can't take grapefruit if you're on there. So there are lots of really good examples of either, nutrients in diet that aren't good or are good, right? Yeah, yeah.
Great.
Yeah.
Well, I think we're over the time. I'm pretty sure next
Okay.
time you should give us a half an hour for discussion. But it's very interesting topic. Thank you so much, Susan.
Okay. Thank you.
All right. Bye.
Bye,
Bye.
Susan.